पोस्टर में भी आप ही करेंगे अगर आपके पास हैं तो, और अगर आपने प्रोवाइड किए तो यू मे आस्क, बट बेटर यही होगा कि आप करें. सॉरी. ओके. Good morning, everyone, and good morning, Mr. Justin Mohan, Dr. T.K. Srivastava. Good morning. How are you, sir? How are you doing?
Very much fine. Yes.
Okay. Thanks for coming. I was thinking that perhaps your meeting will keep you engaged.
Actually, I am going to Montreal now. In another 2 hours, I am leaving. But since I have given word for you, I have accordingly planned. I just
Thank you
for
so
some
much.
time.
Thanks a lot. Thanks a lot. Yeah, it's early in the morning, and we see there are very little users. Hopefully, the people will join very soon.
Okay.
We had a fantastic conference yesterday. Just to update you, more than 300 registrants and people, and the whole university from KSN University of Agriculture, Horticulture Sciences, Shimoga, had joined physically. Today they will not join physically, but they will be online people from the university and others also. So in today's session, sir, we have you as the chairman of the session, and along with you, we have two co-chairmen. Dr. T.K. Srivastava is from the Indian Institute of Sugarcane Research. He's a professor there, and renowned professor of sugarcane. And along with him, we have another co-chairman, chairperson, Dr. Lakshmi Hegde is there from Shivmoga.
Good morning, sir.
Dr. Jay Lakshmi Narayan Hegde.
Yeah,
Good morning. Good morning, Dr. Bharadwaj and everybody.
So good morning, everybody. Good morning.
I'm T.K. Srivastava from Lucknow.
Yes, sir.
good morning.
And
Sir.
along with that, we have the conveners, Dr. Tarun Adak from the Central Institute of Subtropical Horticulture, Lucknow, India, and another convener, two convener, in fact, Dr. Kapil Malik and Dr. Manurani would be there. So they would be joining. Manurani has already joined.
Okay.
So we have two lead speakers in today's. There was one cancellation, and so we had to change the program. But there are two lead speakers. One is Dr. Vasudeva. He's the Dean Forestry in the College of Forestry, Sirsi, UAS, Dharwad, and Karnataka. And Dr. Sanjay Arora. He's the principal scientist at Central Soil Salinity Research Institute and works on the microbes and biodiversity conservation.
Okay.
Two lead speakers, and then followed by 15 oral presentations. And there's a parallel session which is going on another Zoom platform where the poster sessions will be carried out.
Okay. Sir, I have a small request from my side.
Okay.
See, I am going to Montreal now. So I will begin and then be there for some time, and then you may have to permit me to leave.
Okay, that is perfectly fine. Dr. T.K. Srivastava will take over.
Yeah, please, Srivastava sir can take over from me.
Thank you so much, sir. Thank you so much. Thanks for being here. We are really blessed to have you here today.
Yeah.
So we wait for another 1 minute, maybe.
No, you take your time. I am there. I will begin and then I will be for some time and then only leave.
Yeah, so to start the session, I welcome everybody who's present here today, and I hope the other participants will join because we have a very tight schedule for today. We have to carry out 3 sessions before 3 o'clock in the evening before we go for the valedictory session. And I see that Dr. Sanjay Arora is present there. He's the first lead speaker, and also Dr. Vasudeva, I saw he's also present, the second lead speaker for today's session, important session on biodiversity conservation and soil health. And this is the technical session 5. Dr. Vasudeva, good morning. So we, to start the session and for everybody's introduction, we have with us today Mr. J. Justin Mohan. He is IFS and he is the Secretary, National Biodiversity Authority, Taramani, Chennai, India, a very important body regarding the biodiversity conservation. He's been working in the government for the last 21 years in the field of biodiversity conservation, project management, infrastructure development, rehabilitation, development of conflict areas, agriculture, horticulture, groundwater, and forest management. National Biodiversity Authority is involved in the facilitation and regulation of biological resources for commercial use and research, and to implement the provisions of Biological Diversity Act 2002 enacted by Government of India. With that introduction, sir, we are very blessed that you are chairing the session today. I hope the participants would also be very happy, and I hope they join because we have a very distinguished chairman present in today's session, and we have the lead speaker 1 and 2 also present in the session today. Dr. Sanjay Arora is a principal scientist working on the microbial diversity conservation for a long time in the coastal areas and the inland areas. And we have Dr. Vasudeva, he is the Dean, College of Forestry at Sirsi, Karnataka, India. So both the lead speakers are present there. To start with, I request Dr. Sanjay Arora to share his presentation, and so that the chairman could carry out the proceedings. In between, as requested by the chairman, he would be leaving for the Montreal very soon, within 2 hours, and so he may have to leave. And Dr. Srivastava, I would be requesting you to chair the remaining session. That would be grateful for us. That would be really, really good for us. So with that, I hand over the whole session to Mr. Chairman for carrying out the further proceedings. Thank you, sir.
Thank you very much, sir. Thank you very much for inviting us to this conference. I am very happy that this topic has also been included in this conference, that is soil biodiversity, because as you know, it's a very, very important component of our biodiversity. Soil health is very, very important for increasing our agricultural production and productivity. And the Conference of Parties of the Convention on Biological Diversity, which is just starting tomorrow, will also deliberate on this issue. In fact, there have been 22 targets for the global community, and one target to conserve our biodiversity is to preserve our soil health. And I am so happy that this is also being deliberated in this conference. I would also be part of that CBD conference in Montreal, for which I will be leaving today. As I speak, also today, this Honorable Chief Minister of Kerala would be honoring the Soil Survey and Soil Conservation Department for coming up with the mobile app for the farmers. So they are giving an award for best department for mobile governance. Because as you know, in 2015, the government introduced the soil health card scheme, and almost all states implemented that scheme, but Kerala was one state which went one step forward and came up with a mobile app based on the information collected as part of the soil health card scheme, and that helped the farmers of Kerala to actually download the app on their Android platform and then find out for which crop, what is the optimum manure application to be applied. So at that point of time, I was the director of the department, and 1 year after the project was implemented, we had a experiment done in Kuttanad, where paddy is cultivated below sea level in Kerala, to find out whether this scheme was really effective. And to our surprise, we did a proper statistical experiment, and to our surprise, we found that there was a 10% yield when a farmer applies an optimum amount of manure. So, and also there was a 30% reduction in cost of cultivation by reducing the fertilizer application. So that went a long way to convince the farmers of the state to go in for an optimum manure application, so that the soil health of the farmlands get preserved. I'm so happy that the government has recognized this effort, and they are giving this award today. And as we are just moving towards the International Soil Day, World Soil Day on 5th December,
Yeah.
I'm sure that the deliberations that are going to happen today, which is also being recorded, can be shared to other research institutions and other departments which are involved in agriculture and soil conservation, and also to the National Biodiversity Authority, so that the importance of soil conservation and preserving the soil health, the message on how soil health can improve the production productivity and how it can benefit the farmers, will also reach the policymakers and the bureaucrats who are implementing various schemes and projects. So with this short introduction, I now request Dr. Sanjay Arora, Principal Scientist from the Central Soil Salinity Research Institute, Lucknow, Regional Research Station, Lucknow, to come up and make his presentation. I'm sure that it is going to be a very informative presentation. I will also be there to hear this presentation. I myself, I am a soil microbiologist. I did, after doing my agriculture, I did microbiology. So this is a very fascinating subject, and I'm sure that all those who are attending this presentation will benefit from the presentation and will be going back with a lot of information. Thank you. I now welcome Dr. Sanjay Arora to go in for a brief presentation. Thank you.
Thank you, sir. Respected chairman, as you are also a soil microbiologist, so we are happy to be means you have being the chairman of this session. So, sir, the topic of today is my presentation is soil microbial diversity management for soil health and crop production under salt stress conditions where the soils are already degraded to a certain extent, and the the load of salts is to an extent that the microbial population is reduced drastically. So I will be discussing on this aspect, how to manage this, as we are aware that more than 8% of the global land is affected by salt, and due to this salt, the crop productivity or the land value means very low. The salt affected soils occur in more than 75 countries, covering 30% of the global agriculture area, especially under irrigation condition. And 60% of the salt affected is under sodic as well as saline sodic conditions. Because this is the problem, means what kind of salinity, sodicity, there is differentiation, because classification of salt affected soil includes sodic as well as saline sodic and saline soil. The management of all these three are different. So just to brief about, as per the sustainable development goals, we have to means move towards the land neutrality by 2030, and we are focusing on that aspect. So we are trying that how these can be managed, because food security, as per the FAO World Soil Day theme, that food begins from soil and life begins from soil. So basically, to feed the whole world, these soils are the degraded one. They have the potential, and if we can we are able to revive these lands, then only we are means uh, move towards the food security for the future generation. As you very well know, that these soils vary in nature from saline to non-saline sodic conditions, and confined to arid and semi-arid subhumid regions. In coastal areas, large area is affected by salinity, and as per FAO, if corrective measures are not taken to manage these saline soils, it may result in 30% land loss in the next 25 years and 50% loss by 2050. So we should be very means concerned about, on when we are celebrating World Soil Day, we should be aware that this is the problem and how it can be approached to manage these. About 6.73 million hectare is salt affected area in India, and which is going to increase by 11 million hectare by 2050 if properly not managed. There are many methods to manage these salt affected soil to get higher production from these soils, but these are either costly or not feasible for the farmers, not adaptable by the farmers, or not available in the local market sometimes. But the cheap and eco-friendly approach is use of useful soil microbes, because there is problem of salt stress. Along with that, there is problem of nutrient availability. There is problem of rhizosphere accumulation of salt, and all that can be managed through microorganisms that are already present in soil, but under stress condition, their population drastically reduce. This is the extent and nature of salt affected soils in India. almost in Uttar Pradesh, Punjab, Haryana, Gujarat, coastal salinity is there. In West Bengal, coastal salinity, Tamil Nadu. In coastal belt, we can see from Gujarat to
I'll come.
So this is the physical features. We can see there is a difference between saline as well as sodic condition. The salt accumulation on the surface in 5 to 7 centimeter soil, the salt move up with the with the capillary action, and the rhizospheric condition deteriorates, and the plant growth is affected. And how the pH is related or the salinity is related to means nutrient availability, as it is very clear that most of the nutrients are available in the normal range, or we can say towards neutral. But as the pH increases, the availability reduces, and in sodic condition, the microbial activity is reduced. Availability of nutrients depends upon the microbial population in the soil. Conversion of one form to other is mediated through microbes. Main aim of reclamation of sodic soil is replacement of exchangeable sodium and calcium. For this, gypsum is being considered as the best and the cheapest source. But these days, the availability of gypsum is a question. The purity of gypsum is a problem. It is not easily available in the market. More amount is required in those areas where the reserves are not there. So transportation cost enhances. And there are no other calcium amendment source that can help in reclamation of sodic soil. These days, we are not using SSP. We are more concentrating on the DAP, and this also has resulted in less calcium availability in the soils and more sodium accumulation occurring. So how to means whatever the gypsum is available, how to rationalize the quantity of gypsum. For that, we have developed a decision support system. It is a GIPCAL kind of thing, where on the basis of soil pH, we can estimate the gypsum requirement, because sometimes the availability of gypsum is such low that the farmers underuse it, or sometimes there is ample quantity and they overuse it. So judicious use can be done through this means application. It is we have also developed a mobile app of this, and it is available on Google Play Store, and more than 2,000 users are using it for estimating the quantity of right quantity of gypsum, so that they can get the idea how much quantity of gypsum is required to reclaim their sodic soil. Otherwise, if the government agencies provide gypsum, sometimes farmers get huge quantities and sometimes low quantities due to lack of gypsum requirement estimation technologies available in the state government run laboratories, or there are no facilities for in the laboratories to estimate gypsum requirement. So this can be the easy approach, where you have to put the on the basis of soil health card, as Dr. Justin already told, that government has initiated soil health card scheme, and on that basis, farmer can use this soil health card to estimate the right quantity of gypsum. This was the chemical approach, and on the field also, we have developed a small kit. It has been commercialized large scale. It is being used by ICR. It has been approved, and this can be used immediately in the field itself to test the. Is my voice means okay?
Yes, yes.
Yeah, we are able to hear you.
Okay,
We are
okay,
able
thank
to-
you.
Yes.
So GIP kit is one of the small kit that can be used to estimate the right quantity of gypsum. But these are the approaches based on the chemical, and but the problem is that if we are applying large amount of chemicals also, then it is also creating some other problem. Like in Gujarat, in saline sodic condition, application of gypsum or other chemical amendments have resulted in salinity, development of salinity. one problem is improved and the other exists. So we have to balance this can be means how this salinity is affecting the plant, and due to ionic stress, due to osmotic stress, and sometimes excessive toxicity of sodium and chloride, and this disrupts biochemical processes in the plant, and the cell death occurs and plant growth is affected. Soil quality assessment in these sodic conditions through a mobile app as well as through a desktop-based support system we have developed for easy assessment of sodicity in the soil and what is the quality. We have included this based on the soil quality indicators as well as biological indicators as well as weather parameters, because climate of a region or a weather conditions also affect the salinity. As I already told you that in arid and semi-arid conditions it is more aggravated. These are the management issue options that are available for reclaiming the salt affected soil, basically saline soil drainage, leaching with water in saline sodic condition, adding calcium in the form of gypsum or other like pyrites and other things, leaching with good quality water to remove excess salt. Under sodic condition, adding gypsum with deep tillage and leaching with good quality water is the other option. But in the biological approach, one is the crop management. You can manage these. Living with the salt means not removing the salt out of the ecosystem, but we are managing the soil in such a way that we want to cultivate in the existing salt conditions, and for that we have crop management through different salt tolerant varieties of crops. Like in case of paddy we have CSR 10 to CSR 43. In case of wheat also we have certain varieties. In case of mustard, means these are the options available. And some of the multi-purpose plant species under saline and sodic conditions we have identified. Our institute has done extensive work in that aspect, and Dr. Y.P. Singh, our senior vice president, is one of those who have contributed in this aspect to identify certain plant species that having a different tolerance level, and that can be one of the options to regreen or cultivate these lands for economic use. These are the agroforestry options. Medicinal and aromatic plants having salt tolerance can be one of the options. Uh, How to manage these soils? There are certain organic options also: farmyard manure, green manuring, crop residue, compost, home compost or city compost, concentrated organic manure. These are well-known aspects which are available, and water hyacinth as well as cultivation of green manuring crops is the good option to enhance the carbon content of the soil, to manage the soil rhizosphere, to enhance the biological activity in the soil. There are certain byproducts from different sugar mills as well as other factories that can be a good source of carbon in these soils. Biochar nowadays it is one of the options, having the possibility of absorption of excessive sodium on the soil surface, where we are not removing the salt from out of the system, but we are managing these soils in such a way that we enhance the availability of nutrients as well as biological activity to improve the plant growth. Microbes, as I told you, it is very important part how that can be used to manage these stress and for improving the crop production, as our chairman already described, as these have the potential for future to enhance the crop productivity. PGPR, as you must be knowing, that these are the useful organisms having plant growth promoting traits, and they can improve the plant growth. And some of the halophilic plant growth promoters are being used to not only to improve the growth of plant crop productivity as well as to manage the disease and other disorders in the plants. The different mechanism being adopted by the organism. They have the salt tolerance. They provide or they release certain organic acids. Sometimes they helps in production of indole acetic acid and other means important enzymes that are being used by the plant, and they have the direct as well as indirect mechanism like phytohormone production, nitrogen fixation, siderophores, phosphate solubilization, nutrient acquisition, and as well as zinc and potassium solubilization, which is essentially required under salt stress conditions. As I told you, phytohormones include indole acetic acid, cytokinins, ABA, and polyamines. The microbes have the potential to enhance in the rhizospheric area, and the indirect mechanism is the production of antibiotics or HCN or lytic enzyme that helps in biocontrol and pathogen control in the soils and ultimately improving the crop production. As I told you, in sodic condition 8.5 to the pH is above 8.5, and under that condition, the availability of most of the nutrients is reduced, and most of these nutrients are being taken up by the from the soil by the plants. And if these can be managed, they through microbes which have the potential to sustain salt, like through exopolysaccharide production, antioxidative enzymes, or ACC deaminase enzyme. This is one of the important stress ethylene production enzyme, which is an indicator as well as which enables the microbes to sustain as well as the plant to sustain under salt stress condition. Halophilic bacteria has been in very means in recent years it is highlighted in coastal areas under semi-arid and arid conditions, where these have multiple use not only in the bioremediation of salt affected soils, but for biocontrol activity, plant growth promotion, as well as industrial and food processing units are being using these microbes. Bioremediation is the approach we are focusing on at our institute, where we are this is the eco-friendly, less expensive, and easily adaptable approach. In this, halophilic plant growth promoters having efficient stress potential to sustain stress, and at that same time enhances the plant growth promotion under salt stress conditions. Certain mechanism I will not go in detail, but under saline as well as sodic conditions, the microbes have certain not only for their living or for their means sustaining, they require certain salts, but also for plant to grow in convenient environment is provided by these organisms. Alkalophilic bacteria up to 11 pH they are able to sustain, and they have it has been reported by many workers that they helps in plant growth promotion under stress conditions. We are working on that aspect, and we have in coastal areas we have tested for the isolated different organisms. The organism has been tested in the laboratory as well as in field conditions, either for nitrogen fixation, phosphate solubilization. These are the halophilic organisms tested for salt removal, especially in the rhizospheric condition. They have the efficiency to remove salt for their utilization, and we have prepared the bioformulation of these organisms to reintroduce into the soil where these organisms have been reduced. And initially, we have tested in coastal saline conditions, and we have seen that salt-tolerant varieties, along with the introduction or inoculation of these organisms, enhances the crop production. And from the halophyte plants, which are dominant in coastal areas, the rhizosphere, we have isolated certain organisms, and they have the from endophytes also, as well as soil organisms also. We have tested, and we have been working since last 10 years on that aspect, and we have seen that these have the potential not only to improve crop production under saline condition, but under sodic condition also. And this consortia we have developed and being used in Gujarat, Karnataka, Andhra Pradesh, Uttar Pradesh, Bihar, as well as Punjab and Haryana. And we have good results on that, especially in the cotton, wheat, as well as in rice and different crops we have been using. And along with the application of this consortia, we can reduce gypsum or other chemical amendments by 50%. And the cost is also the cost of improving the soil has been reduced to more than 25%. And thus this technology has been approved by ICR and has been commercialized in the form of halophilic bioformulation, halo azo, halo PSB, halo zinc, and we are mass multiplicating it and providing it to the farmers and other stakeholders after a thorough testing at the field as well as KVK levels as well as with the farmers, and we are promoting this at village levels. And this is to how to rejuvenate these salt affected soils where the productivity is very low. And we have seen that the benefit cost ratio has improved from 2.31 to 2.59 in rice and wheat. And the increase in yield application is very easy. Seed treatment or seedling dip or soil application. Some other organisms we are working on the microbial mycorrhiza fungi having potential to enhance water as well as nutrient uptake. And all this consortia we are promoting through different programs to the farmers and for different crops and being validated by the KVKs, and our honorable DG as well as DDG and director have initiated this for approval from ICR, and after approval this has been commercialized and we are promoting for its wider application in the salt affected areas to manage the salt for optimizing crop production. And the promotion is on the way for healthy soil and healthy management of these salt affected lands, and hope these small micro which are tiny in nature but they have the great potential in remediating the salt affected soil and enhancing crop production, and to meet the food requirement for the future generations. This can be a viable option if properly applied and properly managed and properly promoted. This is all. Thank you for giving me the opportunity. I have shared some of my experience regarding bioremediation of salt affected soil through microbes, and this is the approach where we are not removing the salt from out of the system, living with the salt. And if you have any query, I will be happy to respond. Thank you, sir.
Thank you, Dr. Sanjay Arora. It was a very, very enlightening presentation. I'm sure that there would be some doubts or clarifications from the participants. Shall we open it for discussion now, or we should keep it at the end?
So we can keep it at the end. We have the second lead speaker available already, so we can keep.
Is it Dr. Vasudeva?
Yes, sir. I'm here. Hi.
Yeah. Sir, good morning, sir.
Yeah.
Please, I think you are going to make a presentation on status conservation and sustainable management of soil biological diversity in agro ecosystems.
Yes, sir.
Thank you for joining this.
Kindly enable me to share, sir.
Dr. Bharadwaj, please allow him to share the presentation.
Okay.
Yeah.
Okay. Just move.
Yeah.
So we are on time. So I think the speaker also, I think, has limited his presentation to his time.
Yes, sir.
I have a little bit issue here sharing the screen. Just give me a 5 minute, 1 minute, please.
Sir, why don't you WhatsApp him, WhatsApp the PPT to Dr. Bharadwaj, no? So he can.
That is okay. That is okay. We will be able to do it.
Can you just send the presentation to Dr. Bharadwaj? Dr. Vasudeva sir, we are not able to hear you.
Yes, sir, I can send it, but I will send it through Google.
Is it a heavy presentation?
Yeah, because of.
Sir, the host has to just disable, enable the participant to show the
Yeah.
screen, sir.
Yeah,
That's all.
yeah,
It's a simple thing, sir.
It's one step process.
Meanwhile, Dr. Vasudeva, please send the presentation to them so that if they are finding it difficult, they can
Yes, sir.
straight
Sure.
away project and then you can start presenting.
Dr. Vasudev, good morning.
Yeah, good morning. Good morning, Dr. Nagaraj. How are you?
Yeah, fine. Thank you. Nice to see you.
কম্পিউটারটা চলে খারাপ ব্যাটারি নেই হ্যাঁ একটু দরকার এটা হ্যাঁ ল্যাপটপ তো হবে হ্যাঁ দেখা
I have sent the presentation to Ajay Bharadwaj.
একদম পুরো বসা কম্পিউটারটা পুরো ব্যাটারিতে যাবে
না চলে আর কি সেভাবে হয়
ওয়াইফাই লাগাইছে এখন তো ওয়াইফাই বন্ধ হয়ে গেল
Chairman sir, I have sent the proposal, my presentation to Ajay, Dr. Ajay.
Just a moment, I got it. Yeah. Let me unload it.
Once it is presented, you can just start presenting. They will project it on screen and you can start presenting.
Yes. Actually, somehow.
Dr. Tarabdar, please unmute yourself.
The PPT is not opening. There's some problem with the PPT.
Really? You just have to make me co-host. That's it. That would perhaps solve the problem.
Yeah.
One click. Yeah, one click process. Yeah.
Actually, the host has been transferred to another.
Sir, in that case, you will have to make an extempo presentation.
Okay.
Just
Did
see, for 2 minutes, we can just again try, and then otherwise you can just speak on the subject.
Okay. Thank you very much for this wonderful opportunity. My name is Professor Vasudeva. I work as the Dean of Forest College, Sirsi. This is a constituent college of US Dharwad. I take this opportunity to thank the chairperson, secretary to NBA, Dr. Justin Mohan, my fellow scientists, Srivatsala ji.
हाँ एक इंटरनेशनल ये हो रहा है ना
Yeah. और भी आएंगे क्या?
Please mute.
Yeah, actually, the person who is managing this program should mute the other speakers, except the main speaker. Others should be muted.
Yes, sir. Yes, sir. Actually, the host has been transferred to. We are managing from 2 places, and host has been transferred to the other one, and they are not responding.
Okay,
So,
sir. Okay, no problem. No problem.
Just
Dr. Vasudeva can continue. Sir, please.
So thank you very much for this wonderful opportunity. So I'll be speaking on the status, conservation, and sustainable management of soil biological diversity in agroecosystems. I come from a place in the Central Western Ghats. This is in the northern part of Karnataka. And we are in the College of Forestry, Sirsi, which is located right in the Western Ghats. And as everybody knows, there are 4 hotspots of biological diversity in India. One among them is Western Ghats, which is a rolling ribbon of mountains spread along the southern western part of India. And in fact, it is one of the hottest of hotspots of biological diversity, both with respect to the amount of biological diversity it has, and also the kind of threats that is actually being posed to these. And in fact, the Western Ghat is also the world's natural heritage site recognized by the UNESCO. So going to the aspects of what exactly constitutes the soil biological diversity, the official biodiversity definition includes terrestrial, marine, and aquatic ecosystems, both within the species and between the species, as well as the ecosystems in which they are part. So very clearly, this CBD of 1992 clearly recognizes 3 levels of biological diversity. And most of the biological diversity studies today are focused on what is visible, what is above ground. It could be plants, animals, and which is ranging from, let's say, small insects to the butterflies to mammals. But unfortunately, the one that is not visible has not been very much captured. In fact, there is one subcategory of biological diversity recognized world over called the biodiversity for food and agriculture. It is acronymed as BFA. In fact, this BFA is the biological diversity associated with the production systems, such as crop production.
Dr. Vasudeva, may I interrupt? You can share your presentation now. Sorry for the H. You can share your presentation now.
Thank you. Thank you so much.
Sorry for this.
This is great. Okay. Great.
Is it visible?
Yes, it is visible.
Yeah, visible.
Okay, thank you so much. Yeah, I will straight away go. I'll quickly go through. This is where I come from. My College of Forestry is right in the Western Ghats here. I am from the southern part of India and northern part of Karnataka, and it's very nearer to Goa. Those of you who have not seen it. And the Western Ghats, what you see is a wonderful forest here. It's a UNESCO site. In fact, my institute has been recognized as the best in terms of excellence in forestry research by the ICFRE. So I was talking to you about what constitutes the biological diversity. The official biodiversity definition includes terrestrial, marine, and aquatic systems, both of the species and between the species and the ecosystems. So, in fact, most of the studies today are focused on above ground visible biological diversity. But what about the one which is not visible? So this BFA, or the biodiversity for food and agriculture, is a subcategory of biodiversity, which includes the production systems like the crop production, livestock, forestry, fishery, etc. And they include plant, animal, and then forest genetic resources, as well as the associated biological diversity. So in fact, the soil biological diversity contains two systems. One is the variation in soil life and the variation in soil habitats. Variation in soil life can range from a particular gene to a whole community of organisms found in the soil. And the variation in soil habitats can vary from small micro aggregates to the entire landscapes, like the serpentine soils, sodic soils, etc. So in fact, though the taxonomically it's very, very rich, or functionally they are very, very less. Functionally they belong to very few aspects. And what actually constitutes the soil biological diversity components are three. Number one is a microorganism, which can be fungi, bacteria, algae, nematodes, and protozoa. And then mesofauna, which ranges from 0.1 mm to 2 mm in length, which includes species like mites, and then springtails, so on and so forth. And then microfauna, rather macrofauna, includes animals like earthworms, ants, termites, beetles, spiders, scorpions. And you have mammals like the moles. And then predominantly these mammals are insectivorous, and then that's why they have an extremely good potential to control the insect pests. And this biological diversity can be now categorized into functional groups. Number one is as soil ecosystem engineers. As we know, earthworms are the bioengineers. And second one, functional group, is the biological regulators, like the mites or the nematodes. And the third one is the chemical engineers, where you have microbial decomposers. And these organisms are extremely vital for a range of processes that build and maintain the capacity of the soil. And this is very, very essential for supporting the plant growth, regulate the water flow, store the carbon, control the pest, disease, etc. And these functions depend on the complex web of interactions between the functional and taxonomic organisms. If you take a small look at the simplified version of a soil food web, it starts with above ground biomass receiving the sun's energy and giving a lot of organic matter on which the species like bacteria, fungi feed on it, and then the whole food web and the soil ecosystem services starts. Indeed, after the marine ecosystems, it's the soil ecosystem that has a trophic level right from first to the fifth to a very great extent. Hence, the taxonomically and biologically is extremely rich, but unfortunately very poorly explored kind of it. And the contribution of soil biological diversity to the provisioning of ecosystem services is extremely high. As recognized by the Millennium Ecosystem Assessment, there are 4 services supporting provisioning cultural and regulatory. Except for aesthetic and spiritual and air quality regulation, all other provisions are going to be rendered by the soil biological diversity. So whatever that has been highlighted, like the decomposition, soil nutrient cycling, formation, etc., this is primary production of the biological diversity of the soil. And if you look at the typical numbers of soil organisms in a healthy ecosystem, bacteria may not change when you go from agriculture to prairie to forest system, which may range from 100 million to 1 billion. Fungi, in case of a forest, you may have several 100 yards of deciduous forest. And if you go to a coniferous forest, the ectomycorrhizal fungi will be extremely high. several miles it will spread. And in fact, the whole of the above ground biosystem, the ecosystem is going to be largely governed by what is there inside this soil as an ectomycorrhiza in case of the forest. But it reduces very drastically when we come to agricultural field. Only few yards of fungi may be present if you take 1 gram of soil or 1 teaspoon of soil. Similarly, if you look at earthworms, agriculture soils may host 5 to 30, whereas the forest soils may host anywhere between 10 to 50 kind of it. And what are we talking? Are we talking about something very silly? Are we talking about something very not even worth mentioning? What is the economic worth of soil biological diversity? Pimentel and his co-workers way back in 1997 estimated that organic matter cycling and the waste recycling can be of the order of the economic benefit that we are deriving can be order of around $760 billion US. Soil formation is around $25 US. And thereby, if you look at all these services and activity provided by the soil biological diversity, it can be anywhere between $1,200 to $1,500 billion.